So for the final section of this chapter, we'll look at humoral immunity. We've looked at 3 lines of defense. The first line were the exterior barriers like the skin and the mucous membranes. The second line were all the non-specific responses. And now we're looking at the third line of defense, which is immunity. We covered cellular immunity, where cells that contain pathogens are attacked directly. And now we're looking at humoral immunity, where we have a more indirect attack. In this case, B lymphocytes produce antibodies. So humoral immunity is mediated by B lymphocytes. The antibodies will bind to antigens that are found floating around in the blood and tag them for destruction by other means. Whereas cellular immunity attacks the enemy cells directly. Again, we'll have the 3 stages, just like cellular immunity, where we'll recognize, attack, and remember. In the recognition stage, the immunocompetent B cells will recognize the invading particle. The immunocompetent B cells have thousands of cellular receptors for one particular antigen. Activation of the B cells begins when several of these receptors are bound by an antigen. It links them together. This is why some of the smaller particles can't actually be antigens on their own, because they're too small to link several receptors together. Once we link several of these receptors together, the antigen will be pulled in by receptor-mediated endocytosis. The B cell will process and digest the antigen and then link some of the epitopes of these antigens to their MHC2 proteins. Remember, only antigen-presenting cells have MHC2 proteins, and MHC2 proteins only display foreign antigen parts. Usually the B cell response goes no further unless we see that a helper T cell will bind to the complex of the antigen and MHC protein. At this point, the helper T cell will secrete those interleukins, remember signals between white blood cells, to activate other B cells. This triggers clonal selection, which is production of many more B cells by mitosis. It gives rise to an entire battalion of identical B cells that are programmed against the same antigen. Most of these B cells will differentiate into plasma cells. Plasma cells are much larger than the B cells, and they contain an abundance of rough endoplasmic reticulum. That's because they're going to secrete antibodies at a very high rate. Now, antibodies are proteins, and you remember the endoplasmic reticulum is all about transporting proteins out of the cell. They secrete about 2,000 molecules per second during their short lifespan of 4 to 5 days. That's a pretty amazing number. These antibodies then travel through the body into the blood or any other body fluids and bind to the invading particle. The first exposure usually involves antibodies like immunoglobulin M, and later exposure to the same antigen causes activation of immunoglobulin G. Don't worry too much about these different types of antibodies. We'll take a brief look into antibodies and their structure soon. So then we get into the attack phase. The antibodies will bind to the antigen and they render it harmless and tag it for destruction. Some of the B cells will again differentiate into memory cells. Here's some micrographs showing the difference between B cells and then plasma cells. The plasma cells have this high abundance of the rough endoplasmic reticulum. So we've talked a lot about antibodies. Now we should take a closer look at what an antibody is and how it works. Antibodies are also called immunoglobulins, or Igs. They're defensive gamma globulins that are found in the blood plasma, tissue fluids, and body secretions, and in some leukocyte membranes. The Below we have a picture of an antibody monomer. This is the basic structural unit of an antibody, and it's composed of 4 polypeptide chains. And they're linked by disulfide bonds. I don't want you to get too much into the detail of the structure of these antibodies, but recognize that there are 2 larger heavy chains and 2 lighter chains, and they're pretty much identical to each other, except for this yellow region here, the variable regions. And that their antigen binding sites are at the end of these little Y arms. This is where the epitope of an antigen molecule will bind. There are 5 classes of antibodies. Again, I don't want you to get into details of what those 5 classes are, what they're called, and where they come into action. But do recognize that some of them are monomers of this Y shape. Some of them are dimers of this Y shape, and some of them are even bigger. They're pentamers of this Y shape. Just recognize that there are different classes of antibodies. Your textbook gives much more detail, however, I'm not going to hold you responsible for knowing these different classes. Once the antibodies are released by the plasma cells, they're going to use 4 different mechanisms to render the antigen harmless. So this is the attack phase. Neutralization is where the antibodies will mask the pathogenic region of the antigen so it can no longer have its effect. Another means is by complement fixation. Antigens bind to the antibody and change its shape. This will initiate complement binding, and that leads to inflammation, phagocytosis, immune clearance, and cytolysis that we saw in the nonspecific responses. This is the primary defense we have against foreign cells and bacteria after they enter our bodies. Agglutination is where we see the antibody binding multiple enemy cells, immobilizing them and preventing them from spreading through the body. We saw that when we examined red blood cells and did our blood typing exercise. We would see the blood agglutinate if they contained the antigen for which we added specific antibodies. The final way is through precipitation. This is where the antibody binds to the antigen molecules, not cells, but antigen molecules, and creates an antigen-antibody complex. This is the figure down below here. And it precipitates, so it falls out of solution, and then it's phagocytosed by eosinophils. So once we have these things agglutinated or neutralized, macrophages may come along and gobble them up, or eosinophils might come along and gobble them up. When a person is exposed to a particular antigen for the first time, this immune reaction is called the primary response. The appearance of protective antibodies is delayed for about 3 to 6 days, while the naive B cells multiply and differentiate into plasma cells. Those plasma cells then produce the antibodies, and the antibody titer rises for that particular antigen. We'll see that it peaks about 10 days and soon declines. Then we'll see a different kind of antibody level rise. But the IgG is what allows memory. The primary response leaves our immune system with an immune memory for that specific antigen. During the clonal selection, some of the clone become memory B cells. These are found mainly in the germinal centers of the lymph nodes, and they'll sit there until it's necessary for a secondary response. The secondary response is when we're re-exposed to the same antigen. The plasma cells can form within just hours, and the IgG titer rises very sharply, so it peaks within just a few days. There's no need to go through all of the preceding steps. The response is so rapid that there's no chance for the antigen to elicit a noticeable effect on the body, and so we don't see any illness. Low levels of IgM are also secreted but quickly decline, whereas the IgG are going to remain elevated for weeks or years, which confers long-lasting protection. However, the memory doesn't last nearly as long in humoral immunity as it does in cellular immunity. Here you can see the difference in the number of days from the first exposure to production of the antigens in primary response and in secondary response. We can see here, because there's some memory, almost instantly we'll see that secondary response, and antibodies are launched to attack the invading antigen. So again, the best way to get a grip on this immune material is to diagram it. So spend a little time now and go right into the diagramming process. You might begin with this figure of recognition and the production of antibodies, and then continue to show what the antibodies do and how they maintain some memory. Be sure to address the 4 different possible ways that they launch their attacks and how the antigens might be removed from the system. Put this diagram together with your other diagram, and you'll have the majority of your assignment done for this week. Our text also highlights a number of different immune system disorders. These might be great topics for you to present at the end of the semester if you find this interesting. There are a number of different autoimmune disease and immune deficiency diseases. You may choose to highlight AIDS, for example,